Algae harvesting and dewatering technique. Algae has got a wide range of use. Especially, we use the fatty acid portion, that is the unsaturated omega 3 and omega 6 fatty acid, which has got immense health benefits. And also, this type of fatty acids are found in fishes. And so, we see that if anybody who is a vegetarian don't want to eat fish, they can easily have this algae as a diet supplement. So also, apart from this omega 3 and 6 fatty acids, it has also got vitamins, and certain algae has got antibiotic property too. And so we see that pretty much it can be used in pharmaceutical and nutraceutical use, as a food and feed supplement. And algae, one of the most important used these days, this that algae, the lipid content, we know that algae, much of the lipid content can be further increased by reduction of the nitrogen in their culture media. So by various, and also by genetic engineering, that is a bioprospecting, we can modify the algal genes, and so we can produce more and more lipid content. We can increase the lipid content in algae, and that lipid, that is mostly C18, C16 to C18, they are used in the biodiesel production. So we see also algae is used in wastewater treatment. A large variety of algae is used in wastewater treatment. So we see that this algae has got a uh, wide range of use that is beneficial for human. Hello friends, I am Dr. Aintika Das, and already I have made videos on algae culture techniques, which is in the MSc syllabus, both the lab scale as well as the economic scale, open as well as photobioreactor, and micro as well as macro algae culture technique. And I have put the links to those videos in the description box. And today's video will be about various harvesting and dewatering techniques. So let's start the video. So we see, we have already seen in those videos where I made about the algae culture technique, that algae culture is both open in the raceway ponds as well as in the photobioreactor, which is a closed system. So already as because I have discussed those videos, so now I will not go into detail what this raceway pond, this one, or a photobioreactor is. So algae is pretty much, microalgae is cultured in open as well as this closed system. So in this, after algae reaches a certain threshold level, it is harvested. And today I will be discussing about the harvesting technique, which is divided into fourfold, that is the physical method, the chemical method, the biological method, and the magnetic separation method. Physically, we will be discussing the centrifugation technique, the sedimentation technique, and filtration and flotation technique. The chemical, this in the physical methods, this none of the single method are very much used singly. Some most of the time we see that this centrifugation, it is used along with the gravity sedimentation. First, it is centrifugation and sedimentation are used together, or the sedimentation and filtration used together, or the sedimentation is used together with the flotation. So a combination of techniques are used. And in the chemical, we see flocculate. For flocculants, those chemical flocculants are basically those polymers that has multiple charged ions, and this type of polymer can be obtained organically also. Those are the bioflocculants. For example, the algae, bacteria, and fungi, all these, they are the flocculants, biological flocculants. And chemical flocculants are those polymers that has got charge group, for example alum, and this type of chemical flocculants. And for magnetic separation, it is also a very popular technique when magnetic beads are used. And this is a very, as because this is a very fast process. This centrifugation, the physical process of centrifugation, flocculation, and magnetic separation are widely used. So I'll be discussing all this in detail in the coming slides. The first technique in algal harvesting is centrifugation. It is a very common technique in algal harvesting and dewatering, and we know that centrifugation uses the Stokes law, that is the rate of sedimentation. It will be dependent upon density of the algae in the culture media and also the size of the algae. Usually sedimentation is done in a hydrocyclone or a centrifuge. In a hydrocyclone, there is actually not the movement of the container, but the water, it churns, and the algae, it is clustered using the centripetal force. So hydrocyclone, only the water moves, and in the centrifuge, which is here this diagram, this centrifuge, here this aluminium rotator also, this one, the aluminium rotator is present inside it. Aluminium rotator, it also rotates along with the water and algae, and both this hydrocyclone and centrifuge, they are very effective in case of removing or harvesting the microalgae, because macroalgae are usually harvested by a harvester, and not much is needed for their watering, or not much reduction in the volume is needed in case of macroalgae. But here all the harvesting technique I will be discussing about the microalgae, mainly because of very small size as well as high amount of dilution, and also that the algae, the cell wall on the top of say for example this is an algae, and the cell wall contains all the protein, the polysaccharides, lipids, and they have got many carboxyl groups, the amide groups. So all these have a negative charge. Sorry, I've written a positive here. So this will be just a negative charge. So all the entire surface of the algae is surrounded by negative charged groups, that is a carboxyl group, amide group, that is a part of either carbohydrate or protein or lipid portion of the cell wall. So every algae there is having a outer surface negative charge. So this negative charged algae, they will repel each other. So it will just behave like a colloid, and it will be in a suspension, there will not be aggregation. So this for this reason, microalgae harvesting and dewatering, we have developed many techniques just to overcome these three problems, that is dilution, very small size of the microalgae, as well as the surface charge of the microalgae. So for this reason, centrifugation is done, where the centripetal forces are acting on the algal cell. And here we see in this diagram, this is the culture tank for algae, and from that small amount of culture, algal culture is accumulated here, and controlled in a very controlled way. This algae through this pump enters the centrifuge. Now in the centrifuge, after centrifugation at intermediate time, this at this exit flow, the spent up algal media is removed, and the algae, the algal biomass, it is accumulated here inside the centrifuge. So this is also done in industrial scale, also the same type, aluminium or stainless steel. So this was what I hear, it is the stainless steel, but sometime aluminium centrifuges are also used. In the previous slide, that was an aluminium centrifuge. So we see that this centrifugation is a very common technique in algae harvesting, and especially those algae that are very that are used in production of biodiesel, they are harvested by this technique, very efficient technique and very fast technique. But it has got certain disadvantages also. For example, it as because it is using sheer force, so this there is a chance of the surface wall getting disrupted, and so those algae that are used in food, feed, or antibiotic or pharmaceutical, it is not good for technique for those type of algae separation. Also because it is using mechanical force that is coming from some source of energy, so the cost of energy for this centrifugation is not that much low. So it is a bit cost ineffective also, but it is very time efficient because it is a very fast process. Another process of harvesting is sedimentation. So sedimentation, we all know what sedimentation is, and earlier during the initial stages of this harvesting was done only by gravity sedimentation, where the algal cell, they were not subjected to any physical or chemical treatment, but they just sedimented under, they settled down under their own gravity. That is called the gravity sedimentation. Here we are seeing the gravity sedimentation, where different sized algae, microalgae, this is a large size microalgae, this is an intermediate size microalgae, and this container contained bacterial dimension microalgae. And we see that after a period of 4 days, all they have settled, all the algae has settled down. And but the period of 4 days is quite a long, especially in these days where everything is done on a very high speed. So for waiting for 4 days for a process to complete is very time inefficient, and also it will increase the production cost because of this elaborated period of time. So for this gravity sedimentation, it was discarded, and sedimentation with the help of flocculant or any other physical treatment, for example centrifugation, it was usually replaced the gravity sedimentation. But one thing is that while gravity sedimentation did not include any external factors, so those algae that were harvested in to be used in food, feed industry, pharmaceutical, nutraceuticals, they are needed to be harvested by this gravity sedimentation. and addition of this flocculant or physical treatment like centrifugation, it though increased the concentration of algal biomass, and also reduce the time for separation, but they are not that much safe, because this flocculant and flocculant it creates contamination in the algal biomass. By sometimes the flocculants are also very toxic, and this physical treatment, like for example centrifugation, it actually it causes abrasion or wear and tear to the cell wall. And so if we are collecting, say for example, any vitamins, or they might be broken down in this process, physical treatment process. So this gravity sedimentation is good for food, feed, as well as pharmaceutical, nutraceutical use algae. And if we are using algae for creating biodiesel from algae, that is algal biofuel, then this physical and treatment and chemical treatment, that is flocculant, it is very good process and very time efficient. So commonly sedimentation, it is both the marine as well as freshwater microalgae can be separated by this type of sedimentation. And the chemically, we use a lot of flocculants like this aluminium sulfate, mostly aluminium and iron based flocculants are added. And actually what happens, this flocculant, they speed up the process of sedimentation, and the 95% biomass can easily be achieved within only 62 minutes. Only 62 minutes is needed in a chemical flocculation, in a chemical, and we don't have to wait for four days. And the efficiency of any flocculation, it not only depend upon the type of flocculant we are using, but also the species of algae we are using, that is the size of the algae and the concentration of the algal slurry, and also the ionic strength of the suspended fluid. So all these species determine the efficiency, the speed of sedimentation using chemical flocculants. And using physical, that is centrifugation, I discussed in the previous slide, so I'm not discussing this now. Then we come to, that is the chemical flocculation that I was talking in the previous slide. So here actually what happens, we know that the algal cell, it has got negative charges from the carboxyl and amide moiety. So having this negative charges, they repel each other, they behave like a colloid, they remain suspended in the algal slurry that is it is used. So as because they are behaving like a colloid, so they will take a lot of time to settle down normally by sedimentation. So these flocculants are basically they are substances that in water they dissociate and dissociate into various charged units, and these charged unit, they are neutralizing all the negative charge on the surface of the algae. And as a result, two algae that were earlier repelled from each other by this negative charge, so when the flocculant, they form a layer over the neutralized layer over this algal cell. So now they can two algal cell can come close to each other. They can create a microfloc, and two or three microfloc together they form a macrofloc, and in this way they their weight increases, their size increases, they are easily settled down by gravity. So this is about the flocculation, but the flocculant has, because it's a very widely used process. So we should be using flocculant that is non-toxic and also effective in low concentration, and also it should be inexpensive, because that will in the whole algae culture, right from the inoculation of the algal culture to the obtaining the final dry biomass, the most cost ineffective process is this harvesting and dewatering. It is in fact takes 20 to 30% of the entire process. That is why we should be searching for flocculants that are quite inexpensive. So these flocculants, though flocculation can be done on autoflocculant, that is a general intrinsic property of algae also, but we are preferring both chemical and biological flocculant to speed up the process. These are the some of the chemical flocculants here, but of which most commonly used is the aluminium flocculant. And among the biological, if you are using the algae in a wastewater treatment, bacteria and fungi, they are very common flocculant in wastewater treatment. But if we are using the algae for food purpose or feed purpose or nutraceutical, we can use these type of polyacrylamide, polyethylene, and then there will be no problem of microbial contamination. But if we are using the algae for algal biofuel, then we can easily use this chemical flocculants. So this is the process how flocculation occur, and I have discussed it in the earlier slide how it occurs. But it has got certain disadvantages, that is contamination in case of this food based or feed based algae. So there is a contamination problem, both chemical and biological, because chemical can be toxic as well as biological can be pathogenic. So flocculants is addition of flocculant is not permitted in case of we are using the algae for food feed purpose. And also the process of flocculation, it requires a lot of space, and it is cost inefficient due to high price of the flocculant. And the spent of algal slurry that is left, if it is discarded in the in water body, they are very harmful for the fishes and crustaceans. So they can cause loss of those fishes and crustaceans in the water body where this spent up algal slurry is dumped. And one thing I forgot to mention in the flocculation, that is when the flocculant, when in a solution, when the flocculants are added, so then we usually add flocculant by gradual stirring. This very little amount of speed, that is 20 to 30 rpm, this very slight rotation of a rotor, it is just to improve the mixing of the flocculant for easy dispersion of the flocculant in the algal slurry. And this slight rotation, slight agitation of the media while adding the flocculant is very much increases the flocculation speed of in the algal slurry. Now, electroflocculation. Electroflocculation is the most advanced and very high speed flocculation technique, where actually we earlier seen that the flocculants were mostly aluminium or iron based compounds, chemical flocculants, where actually the the positive charged aluminium and iron, it were they were they dissolve in water, they form ionic group and they neutralize the negative charge of the algae. But in the electroflocculation, this anode is made up of this polyvalent metals, that is aluminium and iron. And when electricity passes, this anode is oxidized, and this metallic ions, they are released in the solution. And there, once released in the solution, they neutralize the algal cell and coat it and help in flocculation. But one added advantage is that in and this electrolysis or electroflocculation, electrocoagulation, also the water is getting converted into oxygen and hydrogen bubbles, and these bubbles, they bring this algae to the surface. They help, they pull the algae, they actually bind the algae bind to the surface of the bubble. And the bubble, the if this is the hydrogen bubble, say for example this is the hydrogen bubble, and we have got all the algal flocks surrounding it. And as the bubble rises, this algae is also raised as a layer, and they form a surface. Here we see that they form a surface layer. All the flocks, they are not settled down, but they form a surface layer, and this surface layer can be easily skimmed off. So this is the process of electroflocculation used in the dewatering or thickening of the algal slurry, and this is a very efficient among all the type of flocculation. Filtration. Filtration is a very basic technique that has been used since a long time. And in fact, filtration was started with just a textile filter, where still now also, if we cultivate algae on a community basis, especially in countries like African countries, where just to provide nutritional supplement, spirulina is grown. And there I've seen a video where they were just using this tray drying using just a textile, that is a cloth mesh. And but on community scale, we have been seeing that belt dewatering. So the first filtration material was just a textile filter, and later it was replaced by a filter with diatomaceous earth. And in fact, very small sized microalgae like Sinus desmos were filtered, and even spirulina were filtered using this technique. But later, semi permeable membrane came into the picture, and they improved the efficiency as well as also reduced the time spent on the filtration process. And even it was used successfully on filtering spirulina. And later it was found that this semi permeable membrane filtration the efficiency can be improved by using this pressure, or if it is done, the filtration with a semi permeable membrane done in a vacuum. Under both this condition, the efficiency and also the speed of filtration both increased. And now later we see the use of micro strainers or ultra filters that are used with a vibrating screen. So micro strainer and ultra filter with a vibrating screen are nowadays used, and they have a much higher efficiency, and much in a very high a very low amount of time this can be the entire filtration process can be done. And in fact, there is now a gravity screen filter dewater press, that is this Amco Volute has been using. So using this filtration, we can both filter out the microalgae that are of much larger dimension, as well as bacterial dimension microalgae. So both these two type of microalgae can be easily harvested using this filtration technique. But only one problem with the filtration technique is that in the holes or the pores of the filter, algae gets aggregated and they can plug the holes. They can they can cause clogging of the holes, and which once there is clogging, we need to replace the filter. That increases the cost of filtration. Then we have another very popular, that is the ultrasonication. Mainly ultrasonication is so popular, especially in harvesting, dewatering algae in food and feed industry, to be used in food and food industry, because they uses just sound. No chemical are added, so that there is no problem of contamination. So in ultrasonication, the waves are used actually. Say for example, this is a container. Here the ultrasonic waves are used. So if this is a container with algae, and the waves, we know that waves move in the form of crest and trough, and that is like this it moves. So this algae that are present, and these are the algae, they are suspended in the media. And so over time what happens, this algae that is suspended, they become accumulating here in the nodal region, like a column in the nodal region. So this like this type of column appears. Actually they aggregate as a column in the nodal region of the waves of the ultrasound. So after that, it happens that after some time of sonication, again we see that algae gets only getting condensed in the nodal region only after a certain time period. So by this way, that is use of sound only, without any chemical process, we can get pure algal aggregate. And in this way, this here we are seeing that this is the algae, and after sonication it is getting separated, and it is separated in a single along the nodular region, and this is the spent up algal slurry. So this is how the ultrasonication occurs. So this is an ultrasonic waves, this picture. And there is one great advantage of this sonication is that, as because no chemicals are used, and also it is a very safe to use this process for algae harvest if you are using it in pharmaceutical, nutraceutical, or food and feed industry. And also because there is no membrane, like in filtration we have a membrane. And so in here in filtration, as because there is a membrane, so algae that is getting filtered, they can choke the pores of the membrane and cause fouling. But here there is no such case. And also the problem that we had in centrifugation, that is the outer surface or having getting damaged by the shear force, that also is not there. And also, as because it does not have much of the mechanical, just only a sound resonator, so only a very limited amount of machines required. So it not only reduces cost, but also mechanical failure is we can prevent mechanical failures because few machines are needed, and it is a very continuous operation, and it is also very highly efficient, that we have seen. But one disadvantage is that this high efficient technique, it is also very high energy demanding. So this is the only drawback of ultrasonication. Magnetic separation is a technique, it is a non invasive and very efficient technique. Here in this diagram, we are seeing that in the algal slurry, and in the algal slurry, either magnetic bead or magnetic nanoparticles, they are added, and they attach to the algal cell, and then by using an external magnet, here we see that the algal cell, they form an aggregate very close to the on the magnetic side, and rest of the water or in the slurry can be drained away. This is a very non-invasive technique, but only problem is that the magnetic nanoparticle, they have to be again removed from the algal cell, algal mass, by again a small amount of dilution. So this is the added extra stage that is used in this algal harvest. Otherwise it is very effective, time efficient, and very effective, and also it is non-invasive because it does not it keeps the entire algal cell intact. It does not causes abrasion or breakdown of the algal cell. So this is a very non-invasive and very time efficient, but also very expensive process. Now we see dissolved air flotation, where we see that if this is an algal cell, and it has got negative charges, but and so repel each other, but these algal cell, they can hitchhike on any large bubble, and they can rise to the top and form a surface layer. Say for example, here in this diagram, inside the algal slurry, air is getting injected, and the bubbles, they attach to any algae, and they rise to the top as a surface layer of scum, and this entire surface layer of algae is formed, which rises to the top because they are getting attached to the air bubble. So this can be this can be done by just addition of bubble, or they can be done by additional using of flocculants, chemical or biological flocculant. While introducing the bubble, this flocculant also introduced, and a little bit of agitation is done by the bubbles that is getting introduced. So the flocculant also mixed well, and the bubble also attaches to the flocks that are formed due to the addition of flocculants. The algae no more attach to the bubble as a single algae, but they attach to the bubble as algal flocks, and they help in quicker removal of algae as a surface scum layer. And this flocculation can also be done, this flocculation and air flotation together can be done with electricity also, that I mentioned, that is a electro flocculation done along with this flotation, where just in addition to the all the process that were done in electro coagulation, one addition step, that is addition of bubble, is done. So at this during the entire harvest process, we are seeing that many 2 or 3 processes, they are used in a sequence or used together to improve the biomass concentration. And so the air flotation process, the the air or bubble, they are introduced either by these 5 processes, that is, either is this solution, there is a reduction of pressure, and the gas that is inside the solution, they gets dispersed, and then we have this that is gas and liquid, they are mechanically mixed together, they are churned together to induce the bubble formation, or the gas is directly injected into the fluid by means of a sparger that occurs in the bioreactors very often. And then electrolytic, where I discussed that in the anode flocculants are released, and also they in the they have got a 2 electrodes, anode and cathode, and inside the in the electrolysis chamber, the broth is placed, and this oxygen as well as hydrogen bubbles are generated by hydrolysis of air, and these 2 bubbles, oxygen and hydrogen bubble, they attach to the cell of algae, and they pull the algae along with it to the top of the surface of water. So if this is the algal this is the container with the algal slurry, and we have sparged or injected the bubbles. So in any case, whatever be the process of introduction of bubble, the bubble, they will rise to the top, and as it rises to the top, it attaches to the cell of the algae, and it pulls the cell to the surface layer. So another process by which this air gets introduced is by creating a vacuum. So all these 5 process, the end result is that the air bubble, it pulls the algal cell to the surface layer, and the top scum is removed very quickly. This is a very quick, time efficient process. And so the entire harvesting and dewatering process, it is done in 3 process, that is the primary concentration. It is mostly the sedimentation, coagulation and flocculation is done to create a primary concentration of algae, and very low concentration, that is 1 to 3% is obtained from this. Then after doing primary concentration, this pre-step, we do the secondary concentration, where 15 to 30% concentration of algae can be obtained, and usually done by centrifugation and filtration. So then after that, after getting a 30% concentration, we are using the drying process, where we have solar drying, which is a very commonly used process. Then also we have belt drying, which is also quite common, especially in industrial scale. And then we have the spray drying, which is also used very much common in the industrial scale. And after this drying process, we get a final concentration of 90% or more of algae. So after seeing the many industrial scale algae harvesting, I have seen that most of them, they first use flocculation, that is flocculant is added by gentle stirring of the slurry in the flocculant tank, and then after it is getting flocculated, the flocculant is skimmed off, and then it is dried in a roller drum with a gravity screen. So this gravity screen, mainly it speeds up the process of dewatering. Then after getting in a roller drum, then they are further finally dewatered by using a press. And this press, it is a screw press, and it is a screws press. And this the entire process has been this machines this obtained from Transcend and this Amconvolute. These are the two companies that they have a machine that includes this flocculation tank, the roller drum, and this screw press dewatering, all these three in a single machine. The size of the machine can vary, and from very large to even small size also. So various size models can be obtained in this two company, that is Transcend and Amcovolute. So that were the process that is used in Amcovolute and Transcend. But generally many other additional steps are also found in industrial scale process also. It begins with a flocculation pre-step, and then a settling. Then after settling, air flotation can be done, and after air flotation we can do is a centrifugation. Then after phase separation in a centrifuge, after the phase separation in a centrifuge, then we just do the extraction of lipid, and by transesterification, and biodiesel from algae can be obtained. Actually, this slide is mainly dedicated to one of my subscriber who asked me to make videos on algal biodiesel production. So this is the process of algal bio. This is the entire flowchart for biodiesel production. After the algal biomass is used for biodiesel production, the leftover water, it is converted to biogas, that is a methane. And the leftover slurry, that is a sludge matter, it is used as a fertilizer, because that sludge matter, it contains minerals. So it is used as a fertilizer. So this is the entire process of algal biodiesel production. So from this entire process includes the harvesting and dewatering up to this, and this transesterification and hydrogenation. These are the process by which biodiesel, these are the two processes by which biodiesel is produced. And this
So the
this
algal mass,
this two steps,
it
that
is
is the anaerobic
mostly
digestion and sludge,
these are
for
how
various
the
type of
leftover matter is also
biodiesel,
utilized.
methane, for various type of this production of this fuel matter. We see that the process for production is different. For the bioethanol and acetone and butanol, we use a fermentation of the algal biomass. But for bio oil production, we use liquefaction. For biodiesel production, it is transesterification. And for biomethane, that is a biogas, we use anaerobic digestion. We also produce biohydrogen by photobiological process, and by pyrolysis of the biomass, we get the heavy metals. So this process of formation of various type of fuel from algal biomass, that I'll discuss in next few videos in detail. But this video was about harvest and dewatering. So that was all about this algal harvest and dewatering. Next video will be on the algal, how we can obtain biodiesel, bioethanol, or biomethanol. So this type of how extraction process is done. The next videos will be on this topic. And if you have any doubts or you want me to make any new videos on any topic, please do write it in the comment section, because your comments, it encourages me. Also, do your subscription encourages me. And if you have already subscribed, please, thanks for subscription. If you like this video, please share it with your friends, so that it can get more viewership. So thanks again for watching this video.